Howdy, folks. Thanks for checking in to Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. As you can tell by the pictures on the screen, in this notes cast, we're going to be talking about meiosis and sexual reproduction. Of course, it's hard to talk about meiosis without talking about mitosis first. I know we spent a couple videos back talking about mitosis. Go watch them if you need to get more details. But any process that's going to end up with cells that are exactly the same, have the same information in them anyway, is going to be done through mitosis. So these are clones that have the same amount of DNA, the same number of chromosomes, the same amount of genetic information, the same genetic information. Though they might differentiate into different cells, genetically they are identical because they come from the fission of cells. When reproduction is achieved mitotically, it is asexual reproduction. This is done by single-celled eukaryotes like yeasts, which are funguses, protists like paramecia and amoebas. It's also done in some multicellular eukaryotes as well. Simple animals like hydras, as well as plants, though plants can reproduce sexually, they can also reproduce asexually. So what are some advantages of asexual reproduction? Well, for one thing, if your body plan works, if your genes are working, then you know that your offspring are going to work because they have the same genes, the same body plan. Of course, we also know that the environment doesn't stay the same. So if all of the individuals in a population have the same genes and that's not a good fit for the new environment, well, now we have some problems. Now, the rest of us sexually reproducing organisms, we are formed through the fusion of cells. Fission is asexual, fusion is sexual. So the joining of a sperm and an egg cell. So do we make these sperm and egg cells by mitosis? The short answer, of course, is no. If we did, we'd end up with eggs that have 46 chromosomes and sperm that have 46 chromosomes, and then the resulting zygote would have 92 chromosomes. And this just can't be. So this is not going to work. In today's age, when just about every lab in the world has access to CRISPR-Cas9 mutagenic gene editing technology, and heck, anybody with 99 bucks can get their gene sequenced through 23andMe, karyotyping might seem kind of crude, but it still has quite a bit of diagnostic utility. Although when we look at cells like this, it doesn't look like we can get much information from them. What a karyotype is, we take some cells from an individual. Most often this is done through amniocentesis, so for a fetus, withdrawing some amniotic fluid and looking for cells that are in the process of mitosis. We stain them, and it looks kind of like this. Doesn't tell us much until we go ahead and cut those out and then arrange them, literally cut and paste them by size and banding pattern so that we can count up the chromosomes and see how many there are. You can see there's 46 chromosomes, so that's good, and they're arranged in 23 pairs also. We can also look at the last pair of chromosomes here, the sex chromosomes, and see that they are the same size and shape. This is two X chromosomes, and when we're talking about sex determination, not necessarily gender determination, but sex determination, two X chromosomes is a human female karyotype. When we look at this group of chromosomes, it's hard to see any difference from the previous karyotype. But once we cut them out and arrange them by banding pattern and size, we see that, yes, there are 46 chromosomes arranged in 23 pairs, so the number is all on. There aren't any extras or missing. But that last pair, the sex chromosomes, are not the same. They're different banding pattern, heck, they're different sizes. This is the karyotype for the male in humans. The concept of homologous chromosomes is critical to understanding how meiosis works. Now, homologous chromosomes are those paired chromosomes that we saw in the previous slides in the karyotypes that are the same size and shape. They're the same size and shape because they carry matching genes. That means they code for the same inherited characters. They have the same information. That's where homologous actually comes from. Now, I have to put matching in quoting marks because they might not be the exact same genes, but they code for the same character. That means they both code for, say, eye color. Here's what I mean. Here's a very simple cell. It is diploid, and its diploid number is 4. So it has 4 chromosomes arranged in 2 pairs. Here's one pair of single chromosomes. They are homologous because excuse me, though this one here at this locus might be a gene that codes for, say, brown eyes, that is eye color. This one might code for blue eyes. They're homologous because they both code for eye color, but they're not necessarily the same genes on there. Anyway, that makes these two single-stranded homologous chromosomes. And once that cell starts to divide, whether through mitosis or meiosis, those chromosomes, the genes, the DNA, is going to replicate. So we end up with double chromosomes that have the same information on them. What this all means is that in order to make sperm and eggs, cells that are going to fuse to have 46 chromosomes, then those sperm and eggs are going to have half the number of chromosomes that a typical cell has, so that when they fuse, now the cell has the typical number of chromosomes. It's this process of meiosis that's going to produce an egg with 23 chromosomes and a sperm with 23 chromosomes. We call those gametes. Those are going to fuse and form a zygote through fertilization that has the characteristic 46 chromosomes. This means that sexually reproducing organisms like ourselves actually alternate our cellular life cycle between diploid cells and haploid cells. So at first, our chromosome number has to be reduced from a diploid to a haploid cell. So we're going from 2N to N. In humans, that's going from 46 chromosomes to 23 chromosomes. And it's meiosis that's going to achieve this reduction in chromosome number in order to make gametes, those sperm and egg cells. Then fertilization is going to return those haploid numbers back up to diploid, going from N to 2N through fusion. We can look at this sexual reproduction lifestyle starting at a different point. Here, our gametes are already made. They're one copy of chromosomes of each homologous pair of chromosomes. That makes them haploid and 1N. Through fertilization, those individual gametes that have individuals of each homologous pair of chromosomes join. So now the zygote has two copies, has pairs of homologous chromosomes, making it diploid or 2N, and that individual will then go through meiosis again to make the same one copy haploid 1N gametes. We'll run through a quick overview here of meiosis before we get to the specifics. Here's our example cell in interphase. So we have the chromatin. Now we see it in prophase, so we can see that it is indeed the same example cell that we've been talking about with 4 chromosomes arranged in 2 pairs. And here you can see in prophase 1 how the homologous pairs of chromosomes are aligning, same as metaphase where they're lined up across the plate. And during anaphase, looks like we have some different coloration going on, and those things start to separate, and we end up with, through telophase, with 2 cells that are haploid with 2 chromosomes each, but those chromosomes are still double chromosomes. So they have to separate so that once the gametes fuse, they can go through mitosis again, go through G1, S phase, G2, and then mitosis, yada, 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 like we were talking about. So those 2 haploid cells with double chromosomes are going to go through prophase again. They're going to go through metaphase and line up. They're going to separate during anaphase and eventually through telophase to make a total of 4 cells. But those 4 cells are still haploid. They still have 2 chromosomes each, but these are single chromosomes now that are ready to fuse and then go through the cell cycle and then mitosis and on and on and on and on. So we go through IPMAT, PMAT. So it's IPMAT, PMAT. This is just a quick shot of what the folks at the College Board who are running the whole AP show want you to know about meiosis. Take a look. In order to achieve this haploidness of gametes, meiosis is 2 rounds of division stacked on top of one another. It starts out with DNA replication, just like mitosis, and then we go through 1 round of division. The job of the first division is to separate the homologous pairs. The second round of division is to separate the sister chromatids. To get a little more detail in each one of these phases of meiosis, that first step, again, is the duplication of DNA. So here I have a different example cell that has 6 chromosomes. There are 3 homologous pairs of chromosomes. So the first thing that has to happen is those get replicated, so that we end up with 6 still. We still have 6 chromosomes, but they're 6 double chromosomes. Now you might be asking yourself, if we're trying to get cells that have half the number of chromosomes, why even bother with replication in the first place? And meiosis is really built upon the same genetic and cellular machinery that mitosis is built on. So it uses some of the same genes. So the DNA is going to be replicated during the S phase of meiosis, just like it's replicated during the S phase of mitosis. After the DNA is replicated and those single chromosomes become double chromosomes, meiosis 1 needs to separate the homologous pairs of chromosomes. So during prophase, instead of individual chromosomes lining up along the equatorial plate, now the homologous pairs actually join together and form a single structure called a tetrad. This process is called synapsis. And now across that metaphase plate, we have tetrads lining up rather than individual double chromosomes. This way, once telophase takes place, those homologous pairs of chromosomes segregate. So now each resulting cell has one of each homologous pair of chromosomes. They are, albeit double chromosomes, but you still only have one. So right there, right during anaphase 1 and telophase 1, the cell has achieved the halving of the chromosome number. The second round of prophase, metaphase, anaphase, and telophase in meiosis 2 is working to achieve the goal of separating those sister chromatids. So those individual double chromosomes now can line up across the metaphase plate and go ahead and divide each time through telophase and yield now a total of 4 cells, each one of those cells having one sister chromatid from each homologous pair of chromosomes. Since these are all 4 the same size, I know that they are sperm cells. Remember, the goal here is to make gametes. The steps of meiosis, again, look a lot like mitosis. They're using the same machinery. So interphase, prophase, metaphase, anaphase, telophase are working to achieve that reduction division to actually break up the homologous pairs of chromosomes. So that now instead of 46 chromosomes, we have 23. In meiosis 2, that second round of prophase, metaphase, anaphase, and telophase, that's going to separate the double chromosomes so that now we have individual sister chromatids, single chromosomes, one of each homologous pair. It's still haploid, so this is going to look just like mitosis. Well, let's take a gander at what it looks like for realsies in an actual cell. Here's a cell that's going through prophase 1. You can see the pairs of chromosomes here together. And here they are, metaphase 1, all lined up on the equatorial plate. And they go through anaphase, and we have doubled chromosomes on each side here. And now I have 2 secondary sex cells. And you can see they're lined up here along the metaphase, each one of the metaphase plates in metaphase 2. And now I have single chromosomes on each side. So the sister chromatids have separated, and now I end up with 4 little nuclei here for individual haploid nuclei. Here's where genetically things start to get interesting. See, during prophase 1, when we have those tetrads formed during synapsis, the individual legs of the chromosomes of the tetrad, the sister chromatids and non-sister chromatids of that homologous pair, can actually break off and reattach with one another. So we actually get a shuffling of the genes, or alleles, I should say, that are on some of those resulting chromatids. It's good to be able to compare mitosis and meiosis. From the resulting cells, how they might be identical in one and not identical in the other, and diploid in one and haploid in the other, where they line up and when they line up in homologous pairs or not in homologous pairs. That'd be good stuff to know. So when we put all of this stuff together, actually quite literally, we take a little meiosis, make a sperm and an egg cell, make these gametes. Those gametes are going to fuse through fertilization, and then we end up with a zygote that has the right number of chromosomes for an entire organism, for the whole organism, in this case 46. And then through mitosis, we're going to get more 46 chromosome cells, and they're going to start to look different and look differentiated from one another and specialized until baby comes. Right, and so the process is meiosis, and then fertilization, and then mitosis and development. Genetically speaking, sex is fabulous. It produces a level of genetic variation that mitosis just can't hold a candle to. This takes place because of genetic recombination, so the fusion of gametes, bringing together alleles that were once separated. So we're recombining those homologous pairs of chromosomes. And through independent assortment of chromosomes, this is which one of the homologous pairs ends up in each gamete when they line up during metaphase 1. Also during prophase 1, we get the mixing of the non-sister chromatid genetic material. And then it's a matter of which sperm is the one, which gamete is the one that fuses with the other gamete in order to create the individual. And because of these three things, you get quite a bit of variation within a population, and it's variation within a population that drives evolution. Let's look a little more specifically at some of the different ways that meiosis achieves this high level of genetic variation within a population. It all comes down to how the gametes of the offspring don't look exactly like, they don't have the same combination of genes as the gametes of the parents. Now, what I mean by this is if we look at homologous pairs of chromosomes in an individual as one of each set of homologs comes from mom, is maternal, and the other set is paternal. When that offspring goes through meiosis, it's not like all the maternal ones line up on one side and the paternal ones line up on the other side, so that you end up with one secondary sex cell with maternal and the other secondary sex cell with paternal. It doesn't work that way. How they line up is random, and so you can see in this diagram here how these two gametes made through meiosis don't look like the gametes from either parent. When we look mathematically at the number of different combinations that can happen through the 23 different chromosomes that humans have, it's pretty staggering. Then when we start throwing in crossing over and the mixing of non-sister chromatids from one position to another, and heck, this doesn't even happen just once on each chromosome, it can happen in multiple places on each chromosome, the combination now becomes limitless. When the variations from independent assortment and the variations from crossing over are compounded with which sperm is the one that is going to fuse with which egg cell, we start to see that you as an individual, unless you have an identical twin, represent the only individual that will ever have your particular combination of alleles, truly an individual. Sexual reproduction certainly creates quite a bit of genetic variability. However, when two parents are creating offspring, each fertilization event is going to have different combinations of gametes, but the parents are painting using the same paint kit. So there's certainly going to be quite a bit of similarity between the individuals. The Followill brothers here of the Kings of Leon, certainly you can see the family resemblance. The Jonas brothers, I see it in their eyes. And here's Charlie Sheen and Emilio Estevez. The resemblance to their dad, Martin Sheen, is certainly striking. Well, that's certainly enough for now. Again, thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. If you have any questions, feel free to send me an email at the email address there on the screen, or just leave one in the comment section below. Other than that, we'll see you in class.